We all dreamt of having strength, agility, speed, like our favorite superheroes, and we know that these individuals got their powers by some genetic mutations. So in real life, is it possible? Maybe genetic engineering could help. Humans were engineering life for thousands of years. Using selective breeding, we strengthened certain useful traits in plants and animals. But with the discovery of DNA, we understood why certain organisms show certain characteristics. Deoxyribonucleic acid is a molecule that carries the genetic instructions used in the growth, development, functioning and reproduction of all known living organisms and many viruses. DNA and RNA are nucleic acids. They are one of the 4 major types of macromolecules that are essential for all known forms of life. The 2 DNA strands are termed polynucleotides since they are composed of simpler monomer units called nucleotides. Each nucleotide is composed of one of 4 nitrogen-containing nucleobases, namely cytosine, guanine, adenine, and thymine. The arrangement of these nucleobases denotes special characteristics in organisms. Genetic engineering, sometimes called genetic modification, is the process of altering the DNA in an organism's genome. This may mean changing one base pair, AT or CG, deleting a whole region of DNA, or introducing an additional copy of a gene. Genetic engineering can be applied to any organism, from a virus to a sheep. It can be used to produce plants that have a higher nutritional value or can tolerate exposure to herbicides. Genetic engineering has been used to produce a type of insulin, very similar to our own, from yeast and bacteria like E. coli. A small piece of circular DNA called a plasmid is extracted from the bacteria or yeast cell. A small section is then cut out of the circular plasmid by restriction enzymes, molecular scissors. The gene for human insulin is inserted into the gap in the plasmid. This plasmid is now genetically modified. The genetically modified plasmid is introduced into a new bacteria or yeast cell. This cell then divides rapidly and starts making insulin. The process of gene engineering is very complicated and required a lot of investment of time and money. Also, the process did not guarantee the required results. But the introduction of CRISPR changed the game. Crispr Cas9 system is the defensive mechanism, used by bacteria to protect themselves against invading pathogens like viruses, much like an immune system. The virus kills most of the bacteria, but few of them survive after being attacked. These surviving bacteria cuts out parts of the virus DNA, and save it in their own genetic code in a DNA archive called CRISPR, where it is saved until it is needed. Next time when the virus attacks, the bacterium quickly makes a RNA copy from the DNA archive and arms a secret weapon, a protein called Cas9. Cas9 scans the virus DNA, and when it finds 100% match, it cuts the virus DNA, thus making it useless and protecting the bacteria. Scientists realized that the system can be programmed. The CRISPR Cas9 system consists of 2 key molecules that introduce a change into the DNA. These are an enzyme called Cas9, acts as a pair of molecular scissors that can cut the 2 strands of DNA at a specific location in the genome, so that bits of DNA can then be added or removed. Secondly, a piece of RNA called guide RNA, consisting of a small piece of pre-designed RNA sequence, which is about 20 bases long, binds to DNA. This pre-designed sequence guides Cas9 to the right part of the genome. This makes sure that the Cas9 enzyme cuts at the right point in the genome. At this stage, the cell recognizes that the DNA is damaged and tries to repair it. Scientists can use the DNA repair machinery to introduce changes to one or more genes in the genome of a cell of interest. CRISPR Cas9 has a lot of potential as a tool for treating a range of medical conditions that have a genetic component, including cancer, hepatitis B, or even high cholesterol. Many of the proposed applications involve editing the genomes of somatic, which is non-reproductive cells, but there has been a lot of interest in and debate about the potential to edit germline, which is reproductive cells. Because any changes made in germline cells will be passed on from generation to generation, it has important ethical implications. Carrying out gene editing in germline cells is currently illegal in most of countries. There is a lot of work focusing on eliminating off-target effects, where the CRISPR Cas9 system cuts at a different gene to the one that was intended to be edited. In most cases, the guide RNA consists of a specific sequence of 20 bases. However, not all 20 bases need to match for the guide RNA to be able to bind. The problem is that, suppose 19 of the 20 complementary bases may exist somewhere completely different in the genome. This means there is potential for the guide RNA to bind there instead of the target sequence. The Cas9 enzyme will then cut at the wrong site and end up introducing a mutation in the wrong location. It could affect a crucial gene or another important part of the genome. What's the future of CRISPR-Cas9? It is likely to be many years before CRISPR-Cas9 is used routinely in humans. US and China has already started using this technique with a view to cure diseases like cancer. Much research is still focusing on its use in animal models or isolated human cells, with the aim to eventually use the technology to routinely treat diseases in humans. Scientists are keen to find a way to ensure that the CRISPR-Cas9 binds and cuts accurately. The two ways in which this may be achieved are: firstly, the designing of better and more specific guide RNAs using our knowledge of the DNA sequence of the genome, and the off-target behavior of different versions of the Cas9 and guide RNA complex. Secondly, the use of a Cas9 enzyme that will only cut a single strand of the target DNA rather than the double strand. This means that two Cas9 enzymes and two guide RNAs have to be in the same place for the cut to be made. This reduces the probability of the cut being made in the wrong place. Thus efforts are made to improve the procedure, and if we succeed, this would change the way in which life works. Have a question? Comment below, or on our Facebook page. Please do like, share, subscribe, and let us know what do you think about this video in the comments below. Thank you for watching. See you soon with another video.